an excellent talk and an exciting study. The next awardee is Susanna Tothova from the Dana-Farber Cancer Institute and Ben Ebert's lab. The title of the abstract is Cohesin Mutations Alter Chromatin Structure, DNA Damage Repair and Splicing, and Offer Novel Therapeutic Vulnerabilities in MDS and AML.
So thank you so much for the award and for the opportunity to present our work at this wonderful meeting. I have no disclosures to make. So today I'm going to tell you about our work that focuses on understanding how one of the protein complexes called the Cohesin complex, shown in this video as a yellow multimeric complex that binds to DNA, facilitates chromatin organization, and how mutations in this complex may lead to transformation in our patients with MDS and AML, and how we could go about targeting them therapeutically. So as many of you know, this complex was initially identified because of its role in sister chromatid cohesion, and more recently, it's been shown to be absolutely critical in organization of chromatin into these large loop structures called topologically associated domains, as well as the enhancer promoter looping, and DNA replication and DNA damage repair. My interest in this complex was really inspired by the observation that mutations in this complex are recurrent not only in myeloid malignancies, but across multiple different cancer types. And so what I'm showing you here is a co-mutation plot from the pan-cancer analysis of the Cancer Genome Atlas data, where we're looking at 12 different cancer types, and different subsets or subunits of the complex and its modulators are shown in rows. Different patients are in each column. And what I hope you can appreciate is that mutations in this complex are not only found across multiple different cancer types, but there are specific features to these mutations. They tend to be mutually exclusive with each other. They're usually predicted loss of function mutations that are heterozygous. And in the context of myeloid malignancies, they represent anywhere from 10 to 20% of patients, and particularly enrichment in the context of myelodysplastic syndrome and secondary AML. STAC2 mutations are the most frequently mutated subunits of the Cohesin complex and represent around 85% of Cohesin mutant cases in MDS. And so, focusing on STAC2 in particular, we decided to answer the question of how these mutations cause transformation and how we could go about therapeutically targeting them by looking at these three key questions. First, we wanted to know whether mutations in this complex lead to any changes in the complex composition or interaction of the complex. Second, we wanted to examine the binding of this complex to chromatin and changes any looping interactions. And finally, we wanted to see if we could identify any specific therapeutic vulnerabilities in the context of Cohesin mutant myeloid malignancies. And again, we'll be focusing on STAC2, but it's really representative of all of the other subunits of Cohesin. And as you already wonderfully heard in Ellie's talk, STAC2 mutations are typical for gene where we see the spread of these predicted loss of function mutations throughout the coding sequence with variable outcomes. And so we actually modeled a number of different mutations that we see in patients that lead to either complete loss of STAC2 protein or truncation of the protein or missplicing of the protein. So to understand what happens to the Cohesin complex when we introduce mutations in AML cell lines, as well as in primary mouse and human cells, we employed immunoprecipitation followed by mass spectrometry. In this experiment, we're using SMC1 as a core subunit of all Cohesin complexes as our bait, and do a pull down in STAC2 wild type versus mutant cells. And the first observation that we made was that there is a switch from STAC2 to STAG1 containing complexes. This switch is associated with a number of changes with regards to the interactome of the wild type and mutant Cohesin complex. We see that the mutant Cohesin complex is now interacting more with members of DNA damage repair machinery and replication proteins, such as RPA proteins and PARP. We see a certain number of lineage-defining transcription factors, including RUNX1, CBP-alpha, and core binding factors that are now preferentially binding to the mutant complex. And there is a large number of RNA binding proteins, including splicing factors, whose interaction is lost as we mutate cohesin and switch to this STAG1 complex. And this is something that we can actually visualize by super resolution microscopy. So in these images, what I'm showing you in green are the different cohesin, staining in wild type and mutant cells. And then in red, we're looking at splicing factor SF3B1, transcription factor RUNX1, or DNA damage repair protein PARP1, and can basically recapitulate the findings we see in our proteomics experiments. So we next wanted to address whether this switch from STAG2 to STAG1 cohesin complexes and the change in the interactome is in any way creating new genetic dependencies or vulnerabilities. And so to address this question, we carried out genome-wide CRISPR screens in cell lines with different mutations of STAG2 that we see in patients versus wild-type cells. And what I'm showing you in this volcano plot is that there, in fact, were a number of hits. So what we're looking at is the differential dependency, where all the genes shown here on the right-hand side, on the left-hand side, are more dependent in the STAG2 mutant setting, and all the hits on the right-hand side are more dependent in the wild-type setting. And STAG1, again, not unexpectedly, came up as one of the top hits. We can show that if you have a single wild-type copy of STAG2 and you knock out STAG1 by CRISPR, nothing happens. But if you have mutated to mutated alleles of STAG2, either a complete loss of the protein or missplicing of the protein, and now you take out STAG1, these cells die. And the reason they die is because now you have unstable karyotypes and abnormal sister chromatid cohesion with railroad chromosomes and premature separation. However, in addition to STAG1, we observed that the same kinds of proteins that we had initially detected in our proteomic studies are now coming up as genetic dependencies. And in fact, when we did an overlap between our proteomic studies and our CRISPR studies, the same lineage-defining transcription factors and RNA-binding proteins and DNA damage and replication machinery were coming up, really highlighting these four pathways as being affected in the context of STAG2 mutations. So we next wanted to address whether this switch from STAG2 to STAG1 and the genetic dependencies that are related to the changes in the interactome of this complex could be in any way explained by differential binding of the complex to chromatin, leading to new looping formation. And so to address this question, we performed ChIP-seq, chromatin immunoprecipitation, followed by sequencing, again using multiple different baits, including SMC1, one of the core subunits of the cohesin complex, in cells that were wild-type versus mutant for STAG2. And so when we performed ChIP-seq, what we observed was that, in fact, there's global decrease in association of chromatin to cohesin complexes, in spite of the fact that STAG1 is now the more preferential subunit that's associated with chromatin. And this was happening in the context of significantly increased acetylated histone marks that are representative of active enhancers and promoters, H3K27 acetyl, and associated with spreading of superenhancers in the STAG2 mutant cells. Now, whether this leads to changes in the actual chromatin structure still remains to be seen. And so we employed the chromatin topology study called Hi-C that allows us to look at these DNA looping interactions genome-wide in cells that are wild-type and mutant for STAG2. And so to briefly summarize this data, when we perform Hi-C, what we observe is that the topologically associated domain boundaries are preserved. However, the insulation score at these boundaries is significantly decreased in the mutant cells versus wild-type cells. And when you have decrease of the insulation, that potentially can lead to changes in the loop extrusion of the loops that I showed you in the beginning video through Cohesin. And in fact, that's the case. And so what we observe is that the loops that are extruded in the STAG2 mutant setting are twice as long as in the wild-type setting, really leading to this model that as we mutate STAG2 and switch from these STAG2 to STAG1 containing complexes, we may have increased processivity of the complex with increased loop extrusion, less insulation, and more mixing between different compartments. We wanted to see whether these changes in looping were associated with gene expression changes and performed RNA sequencing. And in fact, we could see that the significant proportion of the differentially expressed genes are falling into those loops that are now affected by STAC2 loss. And the most significant signatures are related to DNA damage repair and apoptosis. We also looked at splicing in our cells, given the association with splicing factors and the dependency on splicing factors and RNA binding proteins, and observed some, but not significant changes in alternative 3' and 5' splice site variants. So finally, we wanted to see whether we could put it all together and use the dependency on DNA damage repair and replication that we've observed in a therapeutic fashion. We observed in our STAC2 mutant cells that the stability of replication forks was significantly decreased, and we had stalling of replication forks in our model, which led to accumulation of double-stranded DNA breaks, shown here by gamma H2AX staining and immunoblotting, and activation of DNA damage checkpoints. And so we decided to use a PARP inhibitor and trapper called talazoparib to assess whether our STAC2 mutant cells would be more sensitive than compared to the wild-type cells. And in fact, we observed about a 70-fold increase in sensitivity, both in vitro as well as in vivo xenograft models of STAC2 mutant and wild-type cells. And in work that I won't have time to get into the details of, but we'll be happy to discuss afterwards, we observed similar changes when we treat our STAC2 mutant cells with splicing modulators, such as the SF3B1 splicing modulator, H3B8800, both in vitro as well as in vivo. And so in summary, what I've shown you is how we went from really understanding the role of specific STAC2 loss of function mutations that we see in patients from the perspective of the assembly of the cohesin complex and the effect on the interactome of these cohesin complexes. We now know how these mutations lead to changes in chromatin looping in association with specific classes of large protein complexes and processes, in particular related to transcription, replication, DNA damage repair, and splicing. I've shown you how these cells are now showing aberrancies in replication fork stalling, which is leading to accumulation of DNA damage and sensitivity to PARP inhibition, which is a finding we're taking to phase 1b clinical trials that will be opening at our institution in the next couple of weeks in Cohesin mutant MDS AML patients who will be treated with talazoparib. I also briefly mentioned how changes in the looping architecture of STAC2 mutant cells lead to transcriptional changes and altered splicing and sensitivity to splicing modulation, which we're currently pursuing in my laboratory. And so I wanted to thank everybody who's been involved. This work was started in Ben Ebert's lab when I was a postdoctoral fellow in his lab. I have since started my own lab at the Dana-Farber Cancer Institute, and we've worked with wonderful collaborators at the Whitehead Institute, Broad Institute, Dana-Farber, UMass Medical, H3B, and Leeds University. Thank you very much.
Thank you very much.